Hello and welcome. Thank you very much for joining us today for this webinar. I'm Emily Watts. I'm a respiratory consultant and a junior PI at the University of Edinburgh. The hope is that with our fantastic lineup of speakers today, we can highlight how to be strategic in your research and grant applications, and in turn have greater impact and success with our work. We're going to have 4 sessions today, and we'll have time for questions after each session. So please, if you have a question, pop it in the chat during the talk and we'll be able to cover those at the end. So without further delay, I'm delighted to introduce our first speaker, Professor Pietro Fratta, who's a professor of cellular and molecular neuroscience at UCL, Department of Neuromuscular Diseases. Professor Fratta is a neurologist and a clinician scientist, and his work focuses on neurodegenerative diseases. He's going to talk to us about strategies for translating basic science into early translation. So thank you very much.
Hi, Emily, thank you. So yeah, I was asked to kind of an overview of our research with a focus on how this has ended up at least trying to do some translation, right? And I guess the premise is that as a clinician scientist, yes, my lab is really disease focused, and the disease I focus on is ALS, amyotrophic lateral sclerosis, motor neurone disease, better known in the UK. And so this is, in the category of neurodegenerative diseases, the third most common. So after Alzheimer's disease and Parkinson's, and it affects the upper and lower motor neurons. So they degenerate and basically lose their function. So progressively one loses all muscle weakness, walking, use of hands, speech, swallowing and breathing, really, which is a main determinant of death. And there is no effective treatment. And in the US, it's known by a baseball player, Lou Gehrig. And this is not a survival graph here; it's a sport performance graph. It's his batting average. And I think though it's quite important to see that, and this is far from being atypical. He was up there. He was, of course, highly performing. And all of a sudden, right, the course of a year, he's still able to, do professional sports. So probably, much better than any of us here. But then what is really important is what is not in the graph, and the fact that he dies less than 2 years later. So that's how precipitous the disease can be. And the main characteristic of ALS is, or not the main, but definitely the crucial finding that is from 2006 is really that one specific protein called TDP43 was identified to be the hallmark of disease. And so typically TDP43 here in brown in the brain cortex section is in the nucleus of all cells. It's expressed in all cells in the body. But what happens only in disease is that, so these are normal cells, is that it goes out of the nucleus. You see it's depleted. There's just a counter stain there, and it accumulates in the cytoplasm. And actually, to add relevance to this, after it was found in ALS in 2006, it has been since described in other diseases in the brain, like Alzheimer's, and also characterizes a number of myopathies as well, including inclusion body myositis. And so TDP43 binds RNA. It's an RNA binding protein, as illustrated in the complex graph on the left. And it's involved in really all steps of RNA processing. And this includes alternative splicing, which is what we'll be speaking about today. And actually, what is pretty interesting about it is that it doesn't bind just around alternatively spliced exon. It actually binds in a number of introns, like the one depicted here, where actually nothing goes on. There is no alternative splicing. Splicing always goes from this donor to this acceptor. But what was really important and was described in 2015 is the fact that if you take TDP43 away from these normal introns where nothing seems to be happening, actually then there's a sequence that we call cryptic exon that is de-repressed and it starts to be included in the transcript. So you have an extra part of RNA, and it's not conserved, it's pretty erratic, and so typically is either out of frame or contains stop codon, and so leads to RNA degradation. And this is an RNA-seq trace from the original paper describing it in 2015, where you can see on top is a normal HeLa line, I think, and actually, this was, I think, a mouse ES line, but anyways, it doesn't really matter. What it matters is that in the knockout version, which is in red, there's an extra trace, which means an extra exon appearing on the RNA-seq. And so, in the lab, we've basically focused, the lab has become an RNA kind of biology lab in the context of ALS. And so we focused for years on alternative splicing in the context of TDP and ALS, right? And just to say that, this phenomenon was, TDP was recognized to be a crucial player, but, the relevance, the translational relevance of all this was thought to be quite dubious. This science paper from 2015, of course, was of high impact, but wasn't really followed up much clinically for quite a while, just to highlight how it may take some time for errors to kind of emerge. And at least what started to really get us, we were of course interested in the phenomenon of cryptic splicing a lot. It's fun to study under a perspective of RNA biology, but what started to give us some sense of the relevance was the fact that in 2019, two parallel groups described a cryptic exon. There's many of these, right? But in a gene called stathmin2, you can see normally you have exons 1, 2, 3, 4, 5. In this case, you get this extra exon here called, you could call it 2A, where it then polyadenylates. And what really struck us was the fact that when you started to look for this cryptic exon in available collections of hundreds of ALS and controlled brains, you could detect it in ALS, but you could detect it specifically in the spinal cord and the cortex where the disease occurred. And you can see even in ALS, in the cerebellum, when there is no pathology, there's no presence of the cryptic exon. And so, as specific as it can be in the diagram I showed in the face before, in a cell line, this really gave us a sense of how specific it can be, how black and white it can be in patients as well. And so that led on to an increasing amount of work that we did, which I'm not really going to talk about today. It's still translational, but it has to do with biomarkers, which is trying to understand if, usually I told you there's stop codons, but of course, sometimes by chance, these cryptic exons are multiples of 3 and they don't contain stop codons. So they're predicted to lead to the translation of new peptides. And so that's what we've been actually pursuing. And this is published work from us and others as indicated below. And we've basically identified these cryptic peptides to be present in the CSF of patients. And I think there's still a lot of work to do to establish them as biomarkers, but it's definitely a really interesting direction. And the lab has continued to look at general biology of these cryptic phenomena. We haven't stopped looking at pure biology, and we've recently actually found that sometimes when you lack TDP, what you can get is that the UTR of genes gets extended, and actually this leads to increased translation of normal protein. So basically this phenomenon could give rise to loss of protein, increase of protein, or the formation of novel protein. But to get to the translation part, it was still quite a long way. There was actually some doubt that this phenomenon that happens when the cell loses all of TDP was actually going to be of some relevance. But again, when we started, generating really good data sets, that was quite key in iPSC-derived neurons with TDP knockdown. We identified hundreds of points. Each one represents a cryptic splice junction as identified in this volcano plot. But we were really struck by one because of its name. It was called UNC13A. And as I'll show you in a few slides, it was very well known to the ALS community for its genetic but unclear link. And what the trace represents here is that again, it's in iPS neurons, it's really black and white. It never happens here. It appears when you knock down TDP. You can visualize it by FISH, and in cells, iPSC neurons, you just see the white dots in the knockdown. You never see it in controls. It appears in the large-scale data set specifically in ALS and not in controls. And basically, you lose TDP, you lose UNC13A. And the reason it was really interesting is that it was long known that it was the second strongest hit in GWAS. So there's 5 SNPs driving this GWAS hit, and actually 2 were in the intron where the cryptic exon occur, and 1 was actually on top of the cryptic exon. So nobody had, really understood what these SNPs were doing and what was happening to UNC13A. And because, of course, we'd always been looking at UNC13A in a normal condition. But if you look in the context of of TDP knockdown, which is what happened in disease, it all became actually quite instantly clear. And so what really happens is that whatever SNPs you carry, whether they're the risk ones or the non-risk ones, you still get the cryptic exon. But if you have one allele of the risk or homozygote, you get more of it. And I won't go through the details of the paper, but the model we came up with is the following: that in health, TDP is there and the cryptic exon is never included, but in disease you always get the cryptic exon. But if you have the risk or the protective SNP, you basically get to the point of which you have the cryptic exon sooner. And just this slight modulation in how much of this you get determines if you're going to get a faster ALS or a slower ALS, which was actually quite impressive. And so, of course, the first question was, well, if naturally the amount of cryptic exon determines disease progression, actually what happens if I revert this risk to the protective? Or actually, even better, what happens if I totally abolish the cryptic exons from ALS patients? And so, based on this, we actually, and I showed this, but I was asked to kind of go through the translational process. We did, this was 2020, file a patent on this. And we then started working to, back up to date on the patent. And we kind of took a step away from therapeutics and say, well, let's actually totally delete the region of the cryptic exon. Let's generate cells where the cryptic exon cannot happen. So you can induce TDP loss of function, but with no cryptic exon. And we saw that indeed, if you do that, you can actually restore the level of UNC13A. So it was telling us that, yes, this was the cause for the loss of UNC13A. And UNC13A is crucial in releasing synaptic vesicles, so in allowing neurons to really communicate amongst each other. And you can see this was old data from mice. If you take it out from mice, you get much less of these spikes, which kind of indicate synaptic vesicle release. And so we were able to show that indeed in control cells and in knockdown, you get a reduction in synaptic transmission. But if you delete this cryptic exon from the genome, you actually get a restoration of the synaptic release. And so that led us to say, well, how can we actually do this in patients, right? Of course, deleting with Cas9 could be an approach, but it's, I think, a bit far away to be considered, or maybe to prime edit some of these sites. These are approaches we are working on. But the simplest thing is, of course, to give antisense oligonucleotides, which are proving to be quite effective in some neurodegenerative diseases. And so we had never developed an ASO in the lab. This was really kind of attempts from PhD students and postdocs to design them, but they actually worked pretty well. And so you can see this is an RT-PCR of a normal cell, and here you get the cryptic, are these high 2 bands. And when you treat with the ASOs, you basically eliminate the cryptic exon and you restore the normal transcript. And so we could show that we could not just restore RNA, we could restore protein. We could restore protein at the synapse, as indicated here. Not much protein at the synapse if you have a control ASO, but if you have the UNC13 ASO, you restore it there. And we could restore function. So as you saw before, less spikes in TDP knockdown. If you give an ASO, you can actually restore neuronal function in these ALS models. And so really the conclusion to all this was that, we, by basically investigating a phenomenon that was kind of, not really thought to be that related to disease, we actually then, ended up uncovering why UNC13A risk SNPs were actually causing an ALS risk. And then we went off to show that TDP43 has impacts on synaptic transmission, and that we could actually rescue all this by rescuing UNC13A either by genomic manipulations or with ASOs. And so, just to illustrate the journey in that on the timeline, it was actually, well, it feels like a long time. I think taking a step back, it was actually quite quick. So we're not aware of this story basically before lockdown. This is a story that came out from our analysis during lockdown, so 2020, which is the same year we filed the patent. Next year, we had the preprint ready and it took a year to publish. And then, so that was the first paper establishing the link. And then the years since 23, 22, 23, 24 were really involved in showing the functional impact of this and the effect of antisense oligonucleotides, which have come to a preprint recently this year. And in all this, of course, this is work that our lab has done fully independently as a lab, collaboration, of course, with other academic institutions. But, now it's at the phase in which, of course, if you want to bring ASOs to patients, it does require much more. And so throughout the years, we've been in contact with US venture capital, and this has now led to, the creation of a startup company, which hopefully will next year or the following deliver ASOs to patients in initial trials. So that's kind of the journey from looking at cryptic splicing to at least trying out something in patients that we hope will have an impact. And so that's the first kind of more conventional thing. But then I think it's or let's say unexpected. And then the second kind of translational example I was going to give was instead a bit more kind of coming from an idea instead of than an erratic finding, and but always thinking about cryptic exons. And so the premise around all of this is the fact that, and this is not just the case for ALS, I think this is probably applies to most of the diseases any of us is studying, is the fact that maybe in vitro, if you have a homogeneous population, there are a number of approaches that can actually cure the cell in a dish. But the problem is that the disease cells are actually just a selected few in the brain in our case. And so this is actually an image that I showed you before, which is probably highlighting an area where there is really abundant pathology, right? But, and so there's in red, diseased neurons, and in green, healthy ones. But actually, if you take a step back and look at the whole CNS, the ratio is much less, right? There's probably very few diseased neurons compared to a whole bunch of normal ones. And so the issue, of course, is that if one takes all these approaches that work really well in the nice setting we create ourselves in vitro with a homogeneous population, and you treat a whole brain, maybe you even limit the expression to neurons, okay? So let's take away liver toxicity. But typically, even if these are, relatively benign things one is giving, with, with sustained expression, one can probably cure, as one does in vitro, the disease cell, but you do cause toxicity to other cells that would be normal. And this, of course, then defeats the point of a therapy, right? And so we thought, well, we'd ideally want a therapy that, sure, administers as gene therapy, goes to all cells, right? But that it just turns on. So all cells have it, but it just turns on when needed, right? So when the pathology happens, and actually, ideally, if you solve the pathology, it then turns off. And so we thought, well, what, could we actually use cryptic exons to do that, right? So what I, tried to explain in the first part of the talk is that when TDP is lacking, these things pop up. And this is what normally happens, and it leads to RNA degradation. But we thought, well, why don't we do something different? Why don't we actually create like a viral gene therapy that does the opposite? So when TDP is there, the splicing of the gene therapy will lead to something which is nonsense. So no RNA degradation, no gene therapy product. But when TDP is lost, so only in the disease cells, will an extra exon be included of our gene therapy? And only in that case will the RNA make sense and be translated to a protein or therapeutic agent. And so, based on that idea, again, a lockdown idea, we started to, develop this. And of course, to start, we didn't start with gene therapies. We started with red fluorescent proteins just to exemplify, right? And so, of course, if you have a constitutive cDNA of a red fluorophore, you're going to get red in cells that have TDP or that lack TDP. But if you actually manage to stick a cryptic exon in the middle of the fluorophore sequence, you actually will get no red fluorescence when TDP is there, because this bit will not be included. And only when TDP is lacking, only when all three exons are, spliced together, will there be red fluorescence. And so I think there is no time to go through how Oscar Wilkins in the lab, now brilliant postdoc there, at the time student, came up with a system to actually design these artificial cryptic exons. But I'll just show you quickly the fact that generally they do seem to work, and actually pretty well. So this is basically cells that represent the diagram I showed you before. So a constitutive red fluorophore, in this case is M scarlet, is expressed in all cells. But if you give it the one with the cryptic exon, indeed, it's expressed only where there's a plus, which means TDP knockdown. So it's really specific in vitro, and actually we also put it in an AV and showed that in this case the expression is M cherry, not M scarlet anymore, but always a red fluorophore, and it works in vivo as well. So when you inject this AV in normal mice on the left, you see no red. In the yellow is TDP, and it marks these large nuclei of the motor neurons. This is a spinal cord cross section. But when you get mice that are that lack TDP just in the motor neurons through Cre recombinase, you now see that these mice start expressing red in the appropriate region. And so this cell here, this is just zooming in, does not have TDP43 and has M cherry, and of course the opposite happens here on the left. So this was really exciting. It works in vivo, and we thought, well, now of course we'd like to put something therapeutic on it, right? And it could be anything, and actually, we hopefully, many people will adopt this to drive their own gene therapies of choice. What we started from is building a chimeric protein, right, that binds the RNA that TDP binds with this yellow part, but doesn't have the C-terminal part of TDP, which is what leads it to aggregate. So it's a protein that was not invented by us, by others, and so it kind of does the same job as TDP, but it doesn't aggregate. But of course, you don't want to put it in all cells. So again, we designed the cryptic exon in the middle of it, and the cryptic exon appears just when you lose TDP. And this is a blot to show you that, the end product works. So of course, the constitutive chimeric protein is always expressed, but the one with this gating system, which we call TDP reg, is expressed only when you lack TDP. And is this useful? Well, it actually is pretty essential. We did a competition assay between cells that express this one, the constitutive one, or the regulated one, and the constitutive ones actually really have a strong disadvantage from the ones that have either the constitutive one or just express a fluorophore, kind of highlighting how something that is not, we're not expressing a caspase here, but should be pretty innocuous, but of course at high levels does cause issues in cells. And so what does this protein do? It should do TDP's job, and does it? Well, we're kind of showing here it does. So UNC13A is the event I went through before. So that one event that we think on its own could be really beneficial to correct in ALS. And you can see that this is the cryptic exon. And when you express either the constitutive version or our own, you really reduce it and you restore the normal UNC13A. That's the same thing as ASOs do. So that's good, but it's just an alternative to the ASOs. But of course, the real strength here is that this binds wherever TDP binds. So it doesn't just correct UNC13A, it corrects the hundreds of other events that TDP43 regulates. And this is an example. We're showing here only 2 other genes. And the example would want to really highlight the fact that some of them, like ARS1, it totally rescues. Some of them is Stathmin2, it rescues, but not fully. So of course, There can be still work to be done in making this chimeric protein better, but it generally has a widespread and good ranging effect. And so this is kind of our second translational idea. So you have a virus, you give it to patients, it goes to all of their neurons, but it sits quiet in the healthy neurons. It does nothing, but instead when the neuron gets diseased, it starts expressing the therapy, which hopefully has an effect. Of course, it's great for toxicity in patients, but there are cases in ALS where people have familial forms, they carry a mutation. In some cases, they're pretty penetrant, and you're going to get the disease at some point or have a very strong chance of doing so. And so this could even allow for kind of preemptive therapies so that as soon as the disease happens, even before it becomes clinically evident, the gene therapy starts having its action only where and when it is needed. And I think I'm exactly on the 25 minutes mark, so I'll just thank the funders, but especially the people who've done the work. So really Oscar up here, who had a picture in one of the slides, responsible for all the second part of the story, and Annalie, Pete, and Matt, and also Oscar for the first part on 13A. And thank you for listening.
Fantastic. Thank you so much. That was a really fascinating insight into how you can go from an idea to a therapy in patients. If anyone's got any questions for Pietro, please do put them in the chat. And while we're waiting for that, can I maybe start with one? I'd like to ask you more about the science, but in order to keep it relevant to everybody, I wonder if I can just ask you about intellectual property and patents. And that's something which, as you said, we do all our work in an academic institution and don't have a lot of exposure to. How did you start kind of understanding how to do that? Do you get institutional support? And what's the process for that?
Well, I mean, your institution will be very keen in doing that. So that's one thing they do support, institutions, because if you convince them to do it. So it has a cost to file a patent, and industry files it. It's not a huge cost, but institutions typically are. So the first step is to convince your patent office that it's a good idea, and then everything comes actually pretty naturally. It's a lot of work, but you typically work with lawyers who are great. They're actually scientists and understand everything, and so that happens. And we kind of ran this through the office. We thought, we were a bit reluctant, and one can even have some issues about IP in general. And I think it would be great to have a system that didn't rely on it, but it is important for the subsequent step, unfortunately. So it is important to have it for subsequent funding, even to get grants. Even if you go for a life art grant and you haven't filed your IP, you're really in a disadvantaged position. It shouldn't be that way, but that's how it is right now, unfortunately.
Great, thank you so much. I think in the interest of time, we will move on if that's okay. So thank you very much again, Pietro, for joining us. I'm now delighted to introduce Conor Judge, who's a senior lecturer in applied clinical data analytics at the University of Galway. He's a consultant nephrologist, an electronic and computer engineer, and has a PhD in epidemiology and biostatistics, so covering a huge range of specialties there. And we're delighted that he's going to talk to us about using artificial intelligence, in particular in chronic diseases, to enhance patient care. Following on from Conor, we're going to be joined by Professor Pearse Keane, who's a professor of artificial medical intelligence at UCL Institute of Ophthalmology. We're really excited to have both these talks together. So what we'll do is we'll have both talks and then questions at the end. And again, if you have any questions, please pop them in the chat, either at the end or as we go. And thank you very much, Conor.
Okay, so thanks for the invitation to speak here. I'm going to talk about the use of natural language processing in research. So as mentioned I work half of my time as a nephrologist inside the hospital, and the other half as a senior lecturer in applied clinical data analytics. So first of all, what is natural language processing? Well, I like the IBM definition here, which refers to it as the branch of AI concerned with giving computers the ability to understand text and spoken words in much the same way that human beings can. And where does natural language processing sit in the Venn diagram here? You can see on the left-hand side, we have artificial intelligence. Inside that, we have machine learning. Inside machine learning, we have a type of machine learning using neural networks called deep learning. And the overlap between natural language processing and deep learning is where large language models come in, which have really brought the word AI to the public's attention since November 2022, since the release of ChatGPT. So I'm going to show you an example of us using natural language processing inside the HSE. So the HSE is the Health Service Executive in Ireland. You consider it the equivalent of the NHS in the UK. And this was the first use of transformers for natural language processing inside the HSE, and it was funded through the Department of Public Expenditure through the Public Service Innovation Fund. So in Ireland, in nephrology, we have an electronic health record called EMED, and it's used for every patient that's on hemodialysis or that has a functioning transplant. And in EMED, there's a mixture of structured data and unstructured data. So the structured data would take the form of past medical history, like ICD-10 codes, or it could take the form of medication, or it could take the form of a lab test result. So structured data is very amenable to audit, quality improvement, and research. Now, unfortunately, in some centres in Ireland and nephrology centres in Ireland, they structured data fields aren't entered as much as they should. So we had an example of this recently, where Ireland was asked to be involved in an international clinical trial for IgA nephropathy, and the first question the trialist had is how many patients in Ireland have IgA nephropathy, and there was no way that we could tell because the structured fields we required for that weren't filled in. Then we also have unstructured data, and this is in the form of free text. So this is if a patient comes to a hemodialysis session and the nurse writes a note about them, or they come to the outpatient clinic and the physician writes a note about them. That is unstructured data, and that's not very amenable to doing audit, quality improvement, or research on without very resource intensive manual chart reviews. So we wanted to use natural language processing to read the unstructured free text and extract the structured information that we needed. And we used a model called BERT. So BERT stands for Bidirectional Encoder Representations from Transformer. So this is a technology that's about 5 or 6 years old now, that was developed originally by Google if you've ever used a search engine, the next word prediction that happens when you're typing in your search terms, that's all powered by BERT. So BERT was initially trained on every word and sentence inside Wikipedia, with a simple task of giving an input of a sequence of words. What is the probability of the next word? It also used another thing called masked word prediction. So given a sequence of words in a sentence and masking one of the words, what is the probability of that word? And repeating this across all of Wikipedia gives the model a knowledge of the statistical relationship between different words in sentences, and learns the structure of language from this. But once it has done that, then we can use it for tasks like classification or named entity recognition. So the flow here is that the patient comes in for their hemodialysis session, or they come into an outpatient clinic. The physician writes a note about them. So, dear primary care physician, the patient came to visit me today. They have a background of type 2 diabetes and hypertension. So the BERT model takes that note and it classifies the patient as either having type 2 diabetes, IgA nephropathy, or hypertension. And then that information is taken out and filled back into the structured fields to allow us to do the audit, the quality improvement and research that we want. So this is a very useful tool to use across clinical data sets. The HSE and the NHS is full of seas of unstructured free text. And that was just our grant. So this was a national project where the head of the National Renal Office was involved, Professor Richard Green, the Chief Clinical Information Officer, Professor Ed Carey, the head of Inside Data Analytics, Howard Johnson, the head of the HSE and National Health Intelligence, and then the Health Innovation Hub. So from once we had that done, then we were able to create a new data set inside the HSE integrated information services that would pass through the structured information unchanged, and would pass through the unstructured information through our classifier developed with BERT. As always with projects requiring data from the health service, the machine learning bit is actually the easy bit, and the hard bit is getting it approved. So our data protection impact assessment for this took about 6 months to develop before, and back and forth before it was finally approved. So this is just our data flow diagram. I'm going to skip over now. So just the extra steps we had to do for the data protection impact assessment, it was too much to get this approved across all of the hospital groups. So we limited to just the west coast of Ireland, and we passed it through a system called Text Wash that removed any names from the free text clinical notes. Then the paragraphs were split into sentences, and an example, and then we passed this to clinical annotators who would read the sentence. So this were senior house officers working in the acute medicine unit in Galway. They read 10,000 notes to create sentence and classification pairs. So here's some examples. So no hypos and BS well controlled, BS being an acronym for blood sugars. And so that's a very kind of nuanced sentence that only kind of a clinical person would know. That means the patient doesn't have diabetes. apologies, does have. Then HbA1c was normal. That means they don't have it. And then no hypertension as an acronym, DM as an acronym, MI as an acronym, and CVA as an acronym. So that was classed as the patient doesn't have diabetes. So text wash is a very important step. So this was an open source tool that would find patient names and ages and identifiers inside the text, and would replace it with placeholders before it was passed to the clinical annotators. For clinical annotation, we used another open source tool called Prodigy that we ran inside the HC network that provided a very easy interface for the SHOs to read thousands of notes. So a note would flash up in front of them, and they'd have to click on the classification and go on to the next note. So they went through this very fast from once it was set up. But really, we'd highly recommend using tools like this, because doing annotation through CSV files or through Excel files will be very error prone. So I'm just going to skip past that a second. So our results then, we saved our classifiers up on Hugging Face, which is a repository for saving machine learning and models, and then our code to implement this was all saved on GitHub and is freely available for anyone to use. Just going to skip past this for a second. These are just the results of our classifiers, and we got some attention for this, and one of our fellows is presenting the results of this as an oral presentation at the American Society of Nephrology. Then the next project on a similar team is being led by one of the researchers in the clinical research facility. Dr. Finn Kruer is his name, and what we've developed here is reading free text echo notes and extracting whether the patient has aortic stenosis, aortic regurgitation, and what their LV function is. The reason that we wanted to do this, my fellowship was in Canada, and Ontario have an incredible database from birth to grave, but they don't have any echo information about their population. They have a data set of open access, or sorry, of free text echo reports. So we wanted to unlock the information in that to ask epidemiology questions like what are the echo markers associated with different types of stroke. I skipped off a second, and Finn has created a pipeline now that uses the newer large language models, the open weight large language models like Llama 3, Llama 3.1, Mistral 7, Falcon 7 billion, and Orca 7 billion as well. So all this is running inside the HSE. So no information about patients is sent to cloud providers. And the tools that we use for this is called GPT for All. So two other very quick things. We created a custom GPT last year for matching grant calls to candidates. So this asks you a series of questions like what year did you graduate from your PhD? What's your thematic area of research, and what are your research goals? So I gave an example of 2022 and hypertension, and to run a pragmatic clinical trial. Then for this custom GPT, we have uploaded a lot of the funding agencies like the Health Research Board in Ireland, Wellcome Trust in the United Kingdom, and some of the European grants as well. So then it creates a custom report that matches the grants based on the FAQ and guidance docs to the candidates. And we also have another database we've recently created that has a list of any previous recipients of those grants inside the university to match people up for mentorship. Another example of using chat GPT in research, this was our code that we did a few years ago to do a sample size calculation for a clinical trial, and I pasted the OR code in, and I asked it to convert this to a Shiny app. So a Shiny app is an app that runs inside OR that provides a graphical user interface for the user to input data into the OR code. So you can see a second later, it has produced the code for this. When the code is ran the first time, it doesn't work. So you paste back the error, you run the code again, and it creates this beautiful or shiny app where you can change the baseline mean and standard deviation of what your score is, adjust your dropout rate, adjust your adherence rate, and it gives you your sample size calculation. So this whole or Shiny app was made in 30 seconds, where in the past this would have taken us a few hours to make. Finally, the last thing, I think I have a minute or two, we designed and set up a master's in applied clinical data and analytics. So we've completed 1 year of it. It was launched September of 23, and we just started our second year of it. The idea behind this master's is to teach healthcare workers health research methodology, applied medical stats, and how to code in OR. So to start with the research question, to find a data set and to use data analytics to answer that research question. So we use a spiral curriculum here where concepts are reintroduced multiple times across the 2 semesters, and every time it's reintroduced to layer additional complexity on it. It's a 90 credit ECT master's, and you have to submit a final year thesis as well. We wanted to teach health data analytics the way that that we learn in medical school, where on the same week you're learning the anatomy of the heart, you'll be learning the physiology of the heart, and you'll be learning the drugs that can be used. So applying this to data and analytics on 1 week when you're learning about case control studies in health research methodology, you'll be learning about logistic regression in applied medical stats, and then on the same week you'll be learning the OR code to implement the logistic regression. So this really helps to cement your learning, because you're learning the concepts in context. And we have a large team working on this, and they're all working inside the clinical research facility as well. And that's the end of my presentation, just at 20 minutes there. So I'll just stop sharing. Thanks, Emily.
Thanks so much, Conor. That's really fascinating insight. As I said, we're going to move straight on to Professor Pearse Keane, who we are delighted to have joining us from the UCL Institute of Ophthalmology. His research focuses on retinal disease, and several years ago now, he developed a collaboration with Google DeepMind to develop AI to aid early detection and treatment of retinal disease. So he's going to follow on from Conor and discuss the use of AI and also collaboration with industry partners to really maximize your research effectiveness. Thank you so much.
Thank you, Emily. Emily, thank you so much for the invitation, and thanks also to Katrina Redden for contacting me about this. Let me just share my screen and hopefully do this somewhat okay. I'm going to talk about using AI in clinical research and some lessons from ophthalmology. If anyone would like to get in touch with me, I've got my Twitter handle on the bottom right corner, and I'm always looking for new followers on Twitter, although not that that's quite so important anymore as it used to be. But on the last slide of my presentation, I'll also give my email address. Now, before I get into the specifics of the AI stuff, I thought maybe I'd just give like a super quick tour or a whistle-stop tour of my clinical academic background. So just to tell you, I went to medical school from 1996 to 2002. I was a medical student in University College Dublin, and I really became interested in clinical research from an early stage. And in fact, as a medical student, I spent a summer in the Wilmer Eye Institute in Johns Hopkins working on bionic eyes. So I was interested in ophthalmology, and I was interested in new technologies and innovation from an early stage. Now, so I finished in 2002. I did my internship, and then I did my basic training in Dublin and the south of Ireland, and that was my basic surgical training in ophthalmology. And then it really became apparent to me, or ever more apparent to me, that I wanted to be a clinical academic. I wanted to go and spend some time in a different country. And so I decided I want to go for a couple of years to the US. And so what I did was I looked up the listing of the top hospitals in the US, and particularly looked at the listing of the top ophthalmic hospitals. And so essentially got that top 10 list, went to the websites of all the different hospitals, looked at all the faculty, and emailed people that I was interested, that their research resonated with me, that I liked the look of them, et cetera, et cetera. And long story short, I ended up getting offered to go to the Doheny Eye Institute in Los Angeles, and went to Doheny for 2 years to do a clinical research fellowship. I got fortunate to work with some amazing, up-and-coming stars in ophthalmology, up-and-coming clinical academics. And a lot of the work that I did there really was kind of a grounding for my transition to becoming an independent researcher, my transition to getting involved in AI research, and so on and so forth. Now, my original plan was to consider staying in the United States and applying to do an ophthalmology residency there. But in 2008, I met this Irish guy at a conference in Florida, and he told me about the NIHR, and he told me about these clinical academic pathways in the UK, and he told me that Moorfields Eye Hospital in London was advertising an academic clinical lecturer post. And so just from meeting this guy, it was kind of like a sliding doors moment for me. And I decided I want, Moorfields is a big name. It would be brilliant to come and work in the UK, come and work in London. And so I was successful in getting an NIHR clinical lecturer post. And that gave me 50% clinical time, 50% research time, and allowed me to complete my training. And of course, probably people listening to this are very, very familiar with integrated academic training and the very nice pathways that they have in the UK. And so when I finished my clinical lecturer post, I was successful in getting an NIHR clinician scientist post. And that funded me as a consultant for 5 years and funded my research for 5 years. It's since been changed the name to an NIHR advanced fellowship. And then just to finish the story and take us to the present day, I'm a consultant ophthalmologist in Moorfields, where I specialize in retinal disease. In 2021, I got promoted to be a full professor at University College London in the Institute of Ophthalmology. And then my current funding is from UK Research and Innovation as a Future Leaders Fellow. And the really cool thing about this funding stream is that it's 7 years of funding, and it's particularly aimed at people or applications that are focused on multidisciplinary research and research that's ambitious and research that's at the boundary between academia and industry. And so if anyone is interested in that, or in clinical academic pathways more generally, or the life as a clinical academic, please do drop me an email. I'm always happy to help. So the other thing just to mention is industry. And so I'll come to my AI work in a moment, but just to highlight that I have worked as a consultant 1 day a week, actually, for about 5 years for Google DeepMind. And then more recently, I work with a number of startups, and so in particular work with a startup from ex-Apple employees based in Shoreditch in London, just down the road from Moorfields, called Bitfount, and they do privacy protecting artificial intelligence. So why do we need AI in ophthalmology? Well, I always like to start with why. And so people outside ophthalmology sometimes can have the perception that ophthalmology is kind of like a niche specialty or a small specialty or like a cushy number in some way. And actually couldn't really be further from the truth. So we know that since 2017 in the NHS, that ophthalmology has overtaken orthopedics as the #1 busiest medical specialty in all of the NHS in terms of clinic appointments. In fact, nearly 10% of all clinic appointments in the NHS are for eyes, and that's nearly 10 million appointments per year. And that's something that's risen by 1/3 in the last 5 years. So we're drowning in the number of patients that we need to see. And unfortunately, there are some patients who go blind because of delays in being seen and treated. And so what I believe is that new technology and innovation, and particularly AI, while it's not a panacea, can be something that helps to at least partially address some of these challenges. Now, I'm of course hugely biased, but I think that AI has, relatively speaking, been one of the medical specialties at the forefront of AI-enabled healthcare. There is some objective reasons to say that. For example, in 2018, we had the first ever entirely autonomous AI system to get FDA clearance, and that was for diabetic retinopathy screening. And in fact, in 2020, the second ever autonomous AI system in any medical specialty to get FDA clearance was also for diabetic retinopathy screening. The thing though is, leaving all the hype aside, what's the current state of adoption of these AI systems, in particular, deep learning systems for medical image analysis? Well, there's a very nice article published in the AI journal from the New England Journal of Medicine at the end of last year from a team in Stanford, where they looked at the adoption of medical AI systems through US insurance claims. And thought I'd highlight just 3 things from this, the abstract of this. So the first is that they looked at over 500 medical AI devices that have received some kind of FDA clearance. Of course, many of those would be decision support systems and not necessarily autonomous systems. And many of those would be quite a broad definition of what is an AI medical device. And then they looked at a database of about 11 billion claims, insurance claims, over a 5-year period. And they looked at these CPT claims and compare them to this medical AI device cleared systems. And the upshot was that, so I guess the good news from an ophthalmology perspective is that the second most commonly used was diabetic retinopathy screening. First was a tool that's used for coronary artery disease analysis. I guess the flip side though is that actually those were the only 2 devices that had more than 10,000 uses, and not a lot more than 10,000 uses. Most of the other devices had almost 0 uses or very, very minimal use. And in fact, just for those top 2, it's still just a minimal adoption. It's and most ophthalmologists and most doctors will not have used any sort of medical AI devices in their clinical setting. So hype aside, we're still super, super early in adoption. Now, I'm not a computer scientist and not an engineer, but my kind of entrance into this field really began back in July 2016 when well, it was 2015 when I started it, but 2016 when we put in place a research collaboration agreement between Moorfields and the artificial intelligence company DeepMind. Now, that involved sharing about 1.2 million retrospective, anonymized OCT scans, optical coherence tomography scans with DeepMind. And as you can imagine, that was a very big undertaking from a technical perspective, but also from a governance perspective and a patient public involvement perspective. And, quite rightly, the public and policymakers and others would be wanting to make sure that we've done everything right in terms of information governance and data protection and ethics approvals and all of those things. But I'll come back to that in a moment. So I approached DeepMind in 2015. We put in place a research collaboration agreement in 2016, and it was very exciting then in August 2018 when we published the first results of our work, our proof of concept in the journal Nature Medicine. And it was exciting because it got featured on the cover of Nature Medicine. And then, of course, this being AI, you can't escape the hype around it. And so it was very exciting to, have your work get that sort of attention, particularly because in my career up to date, I put a lot of work into a lot of papers and put my heart and my soul into these papers, and they're probably read by like 5 people. And so to get that kind of attention was cool, but also, to be honest, a little bit awkward because of the hype factor and because of the fact that we haven't saved the sight of millions of people. And this was an early kind of proof of concept. And in fact, just a few days ago, I got a memory on Facebook where it said something like, "Your memory, this paper was released 6 years ago," on Facebook. And the sort of slightly chastening thing for me is that even now, 6 years later, we haven't saved the sight of millions of people, and this system is not yet being used in the real world. But I can answer questions about that, and I'll tell you a little bit more in a moment about that. So essentially, without wanting to bore the non-ophthalmologists who are listening in the details, the system can run on macular OCT scans. And these are the type of things that you get offered whenever you go to an optician or whenever you go to an eye clinic in the hospital. And then the algorithm will delineate any pathology on the scans, and then it will make a referral suggestion, and it will give a different diagnosis probability. So this is an example of a patient with poorly controlled type 2 diabetes, and I don't think you need to be an ophthalmologist to see that this person has diabetic macular edema or waterlogging of the retina. So anyway, we published our paper in Nature Medicine, and then it was cool that the following year, we were asked to do a live demo of what we had created in the BBC Royal Institution Christmas Lectures. It was hosted by Hannah Fry that year. We've got Keane Hughes, who is a clinical academic who works with DeepMind and with Google. And we have Elaine Manna, who's a patient of mine from Moorfields with age-related macular degeneration. And so it was very nice that we were able to do this demo, although I have to say that it was kind of nerve-wracking. And actually, the demo didn't actually work when we tried to do it live. Although if you look it up on BBC iPlayer and you watch the replay, it's not super obvious that we didn't actually get it to work in real life. Now, the next thing that I just wanted to highlight, and I've already touched on, is the importance of doing things right in terms of the ethics and the data protection and the patient public involvement. And so there was an article in the Telegraph in August 2022 about Google being sued for their work with the Royal Free Hospital about acute kidney injury. And I don't know the rights and wrongs of that case, and I don't know what's happened with that case. But what was nice for me is that we saw at the end of the article, there was a paragraph where there was a quote from this guy called Phil Booth, who's the coordinator of a patient privacy group called Med Confidential. He's a real expert in data security. And he said, "It's perfectly possible to carry out open, ethical, responsible science and health research that has real benefits, such as when DeepMind partnered with Moorfields Eye Hospital." And I would attribute the main reason that we were good in this regard was because of transparency and embedding patient public involvement and engagement from the earliest possible stage. So where are we now in 2024 with this? Well, I think I've already kind of given a spoiler for this, but I thought I might tell you a little bit about this guy here, Ken Cukier. Ken is a senior editor at The Economist. He's famous because he wrote the book Big Data and coined that term a couple of decades ago. And so in March 2021, he actually developed loss of vision in his left eye. We were in a lockdown in London at the time. And so he emailed me saying, like, what should I do? And I gave him a call, and I was able to get him to come into Moorfields emergency department, get seen and get treated. So it turns out that subsequently, I found out that when he was checking in at Moorfields, he was asking the receptionist and the nurses and the doctors, where's the world leading AI system that I've heard so much about? And everybody was looking at him like, what are you talking about? You know, you're crazy, you know, et cetera, et cetera. And so a few days after he was seen and treated, he tweeted that AI is a fraud. I went to Moorfields for emergency eye surgery. Instead of DeepMind's cool algorithms diagnosing me and robot surgeons, I had to accept the excellent skill, thoughtful care of human medical staff. What a disappointment. Now, as it turns out, the condition he had is not something that you would diagnose with an OCT scan, so our AI wouldn't have worked. But nevertheless, I think it's a completely fair point that he made, that despite all the hype about what we've been doing, it's still not translated into the real world. So he kindly invited me to come on his podcast and talk about that, his Babbage podcast, if those are interested. And so what are the challenges? Well, I think that most people most clinical academics would sort of, it's pretty obvious. No, you know, we know that the biggest challenge is that there's a big difference between doing a proof of concept on a retrospective data set, but actually you need to have a much higher standard of clinical validation. Maybe you need to do prospective studies, maybe you need to do trials and other things before it can be safely used in patients. And then, of course, I think most people would know that alongside clinical validation, you need to have regulatory clearance, and that costs a lot of time and money and expertise to make that happen. But what I've learned over the last few years, though, is that those are kind of the known knowns, but the kind of really gnarly challenges are around things like institutional permissions, integration with electronic health records or imaging systems, health economics, implementation and adoption, and so on. And then the thing that I would just really emphasize is I think the biggest blocker to widespread adoption of medical AI is business models. So, you know, when I started this work, I had this kind of naive clinician's view that you just focus on big clinical challenges and everything else is going to take care of itself. But what I've come to realize is that really hardly anyone in the world who are building these medical AI devices has cracked the idea of a business model that works. Because the reality is that if you're going to make one of these as a medical device and get regulatory clearance and make it into a product, it's going to cost tens of millions of pounds to do that. And it's going to cost tens of millions of pounds to maintain that as a product in the following years. And so unless you can do that, unless you can make money, that is just not going to happen. So you need to think about the business model. Now, where are we with this? Well, at Moorfields, we're building a new hospital in the King's Cross area. We're already trying to think about flexibility in the design of the hospital so we can take advantage of new patient pathways that we think AI will enable. But really what excites us most is the high street and how we can try and get this AI system so that we can bring specialized expertise out of centers like Moorfields into high street optometrists. Because of course, in ophthalmology, most primary care is not with the GP, it's with the high street opticians and optometrists. And so really we want this to be something that you go to your Specsavers or your Vision Express or your local optometrist and you have this AI system running in that setting. Now, in the last 3, 4, or 5 minutes, I just want to tell you about some other things that have come from this Moorfields DeepMind collaboration and some of the key learnings. So the first is that we, because of this work, we're able to put together a cloud-based data pipeline for aggregation and curation of our data. And the second is that we're able to get a lot of expertise in information governance and data protection and filling out DPIAs and getting ethical approvals and all of those things. And then we were also able to develop a lot of computer science links. So initially, I went to work with DeepMind because I struggled to get, back in 2015, any interest from university collaborators. And of course, back in 2015, there wasn't quite as much widespread expertise in deep learning for medical image analysis, or at least for me as a clinician, it wasn't as evident. But as we published our other work, it became much easier. And I guess as deep learning expertise became a lot more ubiquitous, it became easier for us to build these university links. So what has that expertise, what have those learnings led to? Well, the first is that in 2019, we got funding from Health Data Research UK to create Insight, the health data research hub for eye health. So we got 3 years of funding at a pretty substantial level to try and create a cloud-based data pipeline at Moorfields, and then to try and reconstitute that at University Hospitals Birmingham, and then see if we could expand to other sites in the UK and maybe even around the world in the longer term. So this is a dashboard that we took from our system, and I'm not going to go really into the details of this, but suffice to say that the exciting thing is that we believe that we've created the world's largest ophthalmic imaging bioresource. We have more than 30 million images linked to clinical metadata at Moorfields Eye Hospital alone. And I think that's pretty cool because of various historical reasons. Moorfields is a very big eye hospital, and we have more ophthalmic imaging data than the top centers in the US, the top 3 centers in the US combined. And that's a pretty unusual situation to be in. Now, we combine our data infrastructure with what we think is gold standard governance. We have worked with the Open Data Institute to create an independent data trust advisory board, as well as the charity Action Against AMD. And we do a lot of patient-public involvement and engagement. So when you come to Moorfields, there are screensavers in our waiting rooms that flash up, which talks about how we use routinely collected data for research. They give people links to learn more, links to opt out, etc. We have leaflets that are given out to people in different languages. And so we really want to engage with them around the power of routinely collected data. And so the other 2 things that I want to talk about are some studies that have come from this data infrastructure that are not related to DeepMind and the work that we've done. So the first is a study called the ALSI study. And the ALSI study is, we essentially got patients aged 40 years and older who had imaging done at Moorfields over a 10-year period, and we got permission to link that data to NHS hospital episode statistics, which is essentially a national database that has ICD-10 codes for every inpatient admission, every outpatient appointment, and every A&E attendance. And so the upshot of that is that we know if we have a patient who attended Moorfields and had an eye scan done, and then they move to Liverpool and have a heart attack, or move to Grimsby and develop multiple sclerosis, then we have that information. We can use that data to train AI models for this. In particular, in this emerging field that we've called oculomics, using the eye as a window to the rest of the body. Now, if you're interested in learning more about the cohort, and in particular, how we approached making this linkage and the governance processes that we went through, we've written about it in the BMJ Open. We think this is a really exciting data set. One caveat to the excitement around it is just to flag that it took us 4 years to be able to get the permissions to make this linkage. And in fact, 4 years is pretty good going, because I think most people without our experience in governance processes would probably have struggled to get through this process. So just to say some initial results, we published a paper in JAMA Psychiatry at the start of 2023, showing changes in the retina in patients with schizophrenia. And then at the end of 2023, we published a paper looking at changes in Parkinson's disease. And so we showed changes in the ganglion cell layer and the internuclear layer of the retina, very subtle changes. But the thing that got a lot of media attention in the UK was the fact that we were able to replicate our findings in UK Biobank, and we saw that the changes were present on average 7 years before patients were diagnosed with Parkinson's disease, albeit very subtle changes, only evident on average across the group. So why do I think that this is exciting? Well, as I've emphasized already, for us, it's all about the high street optometry. And so we know that in the NHS, when you turn 40, as I discovered a few years ago, you get a text message on your phone saying, go to your GP for your cardiovascular health check. And I was busy at the time and I didn't follow up on it. In fact, we know that only about 10% of people do it between 40 and 60, but we know that about 80% of people in that age group will go and have an eye test done. And so, you know, there could be big public health implications if we can pick up eye disease from eye exams. And then the last thing, I've probably already talked for far too long, but I just want to talk about this other, briefly mention this other work that has led indirectly from the DeepMind work, which is around creating foundation models in medicine. So The Economist in July 2022 talked about how huge foundation models are turbocharging progress in AI. And foundation models is a term coined from Stanford, which is really just a kind of rebranding of an old concept, but it's this idea that you can use one model, train it on huge amounts of data, and then fine tune it for a range of different downstream tasks with only small amounts of labeled data. For those who are interested, we just published a review article in the British Journal of Ophthalmology about medical foundation models, at least in an ophthalmic context. And where we come in is that we created a foundation model called RetFound, or Retinal Foundation. And essentially, we took a data set of about 1.6 million retinal images from Moorfields. We pre-trained them with a type of self-supervised learning called a masked autoencoder. And then we were able to show that they could be fine-tuned with only tiny amounts of label data for 10 or more different downstream clinical tasks, like predicting heart failure from retinal photographs or predicting glaucoma and so on and so forth. And we're able to show that that this model was more label efficient, more generalizable, and fairer across a range of tasks. Now, we published this then. It was exciting that we published this in September 2023 in Nature. And then I feel quite proud that we were able to make the model code and the model weights, importantly, open source for non-commercial purposes. So we're already starting to see people around the world taking our model and fine-tuning it for their local applications and improving on our model. You know, we think that this is hopefully a big step forward. So just to finish then, my last slide, you know, I know probably there's hardly any ophthalmologists that are listening, but I guess I just wanted to share some lessons from ophthalmology. And I do believe, even accounting for hype, that we can take technologies such as the slit lamp microscope that haven't changed really in more than 100 years, and we can reinvent them for the 21st century using new technologies like artificial intelligence. And then, as promised, that's my email address. Please do get in touch. Thank you very much.
Fantastic. Thank you so much. That was a really fascinating insight into the world of AI, about which I have to say I don't know a huge amount. If you've got questions either for Pearse or for Conor, please do pop them into the chat. And maybe if I can kick off just while people are gathering their thoughts. I get, and as I say, you know, this is very much not my area, but my question for both of you really is, do you think our systems within either the HSE or the NHS are fit for purpose, given how important AI is going to be in the coming years? You know, I remember, you know, you've got one system for radiology, one for letters. Do we need to completely reinvent it, or can we use what we have and design tools that are able to kind of extract the information that we need?
What do you think, Conor?
Yeah, we submitted a grant last year to Science Foundation Ireland to buy a GPU cluster for the HSE. Now, unfortunately, it just fell below the mark, but I think that's what we need. We need on-premises hardware, because we can't be relying on cloud providers. It's not cost effective under a certain amount, and also it adds a lot of complexity with transferring data outside of the HSE or the NHS. Professor Keane, has the NHS any plans for investing in GPU infrastructure in their own data centers, or are they going to go for a kind of a hybrid approach or a kind of a cloud-based approach?
Yeah, that's there's a risk that I could talk for like half an hour on this particular one, and
we could take
I'm
it
happy.
offline.
I'm happy to talk separately, but
Yeah.
I guess generally I'm quite a big proponent of the cloud for NHS data, and I think that in the NHS there was national guidance in 2018 supporting the use of the cloud, and so now the e-referral system for the NHS is on Amazon, and the blood transplant system is on Microsoft Azure, and we've been able to get through the permissions so that we can put Moorfields data on. It's like a Moorfields cloud, but it's Google Cloud Platform who provides the but that doesn't mean that Google get access to the data. It's just they're the people we work with. I think the cloud is great, but as you say, it is super expensive for the longer term. And so I'm coming to realize that you need to have a kind of hybrid approach where ideally you would have some cloud where you could do some like big experiments, or you could more easily quickly generate data sets and other things, but that you probably need some local compute. And some of that should be just workstations, and some of that should be like a local cluster. I think maybe I'll finish because I don't want to bore people with the headaches of it, but one of the challenges is that for example, in UCL, there is some compute, there is a high performance computing clusters, but in order for the data to go from Moorfields to UCL, there has to be a data sharing agreement in place, even though they're kind of like very closely linked institutions, and that is very challenging to get. And so it becomes very difficult. And then separately on the NHS front, there's this big kind of national kind of move towards secure data environments and data not leaving the NHS and all of those types of things, which also makes it harder for the data to go to the university, which I don't think is a good thing. But those secure data environments are, for the most part, very limited in the type of analysis that you can do. And there's a very limited ability to kind of like get lots of GPUs and do cool stuff. But yeah, I kind of went off, Emily. I didn't really answer the other parts of your question, but yeah.
That's great, thank you. So question from Nobby in the chat, really interesting talks. Can you say a bit more about balancing business models versus open source foundational model and weights and how that might work, please?
Well, maybe if it's okay, Conor, I might go first, but I think that one of the biggest challenges that you'll face is commercializing these models. Suppose you published a paper and you now want to commercialize it. One of the challenges that you probably used NHS data and you've used university expertise to develop a model. That means that if you want to form a company or license it to a company, that you're going to need to have NHS approval and university approval. And that means you could easily spend a couple of years of nothing happening because of trying to get those permissions. And so I think ultimately, if you want to get things that are used in clinical care, you probably do have to think about this kind of commercialization type thing. In my experience, I've tried to combine both approaches. So we've been looking for commercialization of the work we've been doing with DeepMind, but then for some of the work we did with the RETFand foundational model, we decided that the best way to have impact was to make it open source and try to have research impact, because that was the quickest way for us to do things.
Brilliant, thank you. And then final question, I think we've got time, from Sandeep is, how do you, for a beginner, and this would include me as well, how does one learn to use ChatGPT?
Is that Conor, would you like to take that one, or?
Yeah, the way to learn it is the way to learn anything else, and it's to interact with it. Like one nice trick, just one kind of takeaway trick is ask it to develop the prompt. So if you describe the thing you want to do, but the output of the first one is the prompt. So you can say I want help to critically appraise our research paper, and I want you to help me to write the prompt to use chat GPT effectively. It will produce a prompt that'll have 20 sentences in it. That's a very complex prompt with lots of nuanced piece of information. Paste that back in, and now you'll have a very useful tool for critically appraising a paper. There's Pearse, you want to join?
No, sorry, I didn't mean to interrupt, but I was just, you gave me a little bit of time to think, so I could answer. But Emily has kindly posted. There's a book that I read called Co-Intelligence, which is from this very good professor from the US called Ethan Mollick. He's a business school professor, and he posts on Twitter and other things about how he uses his ChatGPT. And one of the things he does is he asks his students to use ChatGPT in their assignments. And so I'm trying to get my research group to try and explore the use of ChatGPT or similar LLMs in everything that we do. So we use Claude as well. And I'm like, let's just see where the frontier is in terms of what we can use it. So, you know, if like we were writing a grant recently, and there was like a 6,000 word research plan, and we had just finished that, and then we had to do a 500 word summary. I'm like, let's just put it into an LLM and let's see which is the summary. And then I say to one member of the group, can you use the LLM to create the summary and then tweak it as much as you want? And then another member of the group, can you write a summary without an LLM? And then let's do a blinded test to see who can use it. Right. Or it's just this mindset of incorporating it into our lives. Now you have to have a paid version, because otherwise you can't upload stuff, or you can't upload sensitive stuff. But or you might, like I was doing an interview panel, and for a grant, and I uploaded one of my own NIHR grants to my paid version, and then I said, can you prepare some interview questions based on this application? And it came up with like some pretty good questions, so that you could use it to prepare for interviews and things like that. So it's just about seeing where it works and where it doesn't.
Thanks so much. It was really interesting, Conor, to hear that you're incorporating it into a kind of medical education now. I feel like for those of us who kind of finished university before it came in, we're slightly lost. So it's great to have some resources. I think in the interest of time, we will leave it there. Thank you both so much for that really fantastic insight. And I'm delighted now, actually following on really nicely from Pearse's comments on involving patients and making them understand the importance of their data, to our final session, which is led by Dr. Raahat Manrai, who's a postdoctoral researcher in the Division of Psychiatry here in Edinburgh, and Dr. Amy Ferguson, who's the Network Scientific Coordinator for the Circadian Mental Health Network. We're really delighted to have them join us to give us some insights into patient and public engagement in clinical research, the importance of co-production and lived experience involvement. So thank you both very much.
Yeah, thank you so much for having us. We're excited to talk about this. Just a kind of quick note that we're taking a slightly different track of looking at things. One disclaimer I will have is that, I am not a clinician, so please bear with me. This talk's going to be a little bit different. So basically, I'm going to give you a quick practical guide to what PPIE is all about. So first of all, what is PPIE? You'll have heard this a million times before, but as a reminder, it's patient and public involvement and engagement. So what do we really mean by that? Well, involvement we really think of as actively involving patients, public, those with lived experience of what you're looking at in different aspects of your work. This can include through things like advisory panels, through co-production, which Raahat is going to talk about in the next session, collaborations and things like that. Whereas engagement is more about clearly communicating your work to patients and the public and providing kind of relevant context to what you're doing, to who you are sharing this with. So what we're really going to be focusing on here is the involvement aspect of PPIE, of actively involving those with lived experience in your work. So why is this even important? Well, first of all, it involves the diverse voices in research. It allows them to be included in what's going on when in certain fields, historically, they're not always massively involved. They don't normally have a say in maybe what necessarily is going on. It also gives you access to expertise, a vast wealth of expertise in the thing that you're looking at by allowing people that have lived experience to be involved in what you're doing, as well as providing a kind of real life context for our data. A lot of the time it's all statistics and it's, well, this trend does this, or this is what we see. But what does that really say? Whereas presenting all of that kind of thing and involving people with lived experience gives us a real life context for what we're actually seeing in most cases. Oops, sorry, wrong way. But also now, especially from what you've just heard for previous things, science is so much more diverse and inclusive and things like that than it used to be. And we need all of those different ways to make impacts in what we're doing and to encourage change. So we need as many different viewpoints to look at this and encourage change. And thankfully, a lot of funders and things like that are now seeing this inclusion of PPIE as something super important as well, which is fantastic. So one aspect of PPIE that I'm really going to talk about is more about kind of consultations, advisory groups, and the thing you'll probably hear an awful lot is the word LEAP. So it's a lived experience advisory panel. And so basically it's a group of individuals with a lived experience which provides independent advice based solely on their own personal experience, on their expertise. With that, because you are engaging with people and asking on their expertise based on their own personal lived experience, the way LEAP panels should work is that they are safe and supportive spaces where everyone has an equal voice. There should never be that kind of power dynamic of I am researcher, you are participant. That's not the way things like this should work. Engaging with a LEAP is a two-way relationship. You are asking things of them, but what can you also give back to them where they're giving you their expertise? So there's many different ways that a LEAP could actually be involved in research. So I'm just going to name just a couple. Well, briefly going to name co-production, which I said Raahat is going to talk about in way more detail next. But they can also be involved in things like consultations. So this can even be things of like grant submission. You can have a LEAP involved to tell you what questions should you really be asking. You might have like, yeah, this is my very specific question. I want to know about this very specific condition or this very specific aspect of a condition. But are you asking the right questions about that? Especially, does it matter to the people that live with that? They can also give you great insight into who you should be approaching with all of your kind of questions, and consultations on things like the materials that you use. Will it actually engage with the people that you're wanting to talk to? Will it engage with the general public? Are people even interested in these kind of things? And is what you're using going to do that? As well as collaboration on data that you get, things like that, as well as the actual knowledge exchange part of disseminating what's going on. And that back and forth of they're giving you their expertise and exchanging knowledge for your knowledge as a clinician, as a researcher. So this knowledge exchange can take part again in many different forms. The world is your oyster when it comes to all of this kind of thing. One thing could be the involvement of in the creation of materials, which you'll actually be making publicly available. one example of this that we are currently doing is the Helios BD. So the leap that we work with Helios BD, we are using their experiences. They're providing us with experiences they have of their conditions with bipolar disorder and their experiences of light, of seasons, the different things that they feel and experience, and are creating art through AI generation. So that's one way of doing it. But kind of also through that, they've also helped us kind of interpret data. So they've been involved with interpreting data and that kind of way of providing, again, that real world context. Your LEAP can also be part of helping you disseminate your information, talk about your studies, about what's going on. Within the Circadian Mental Health Network, our LEAP have been really involved with not only things like making sure the actual content is worth looking at and is exciting, but they've also been very, very keen to make sure that it reaches the right people and have been part of disseminating that information as well. You can also include them in, and I would encourage you to include them in conferences, presenting at conferences. So with, again, Helios BD and with Ambient BD, another project that we're working on, we have members of our LEAP panel, the LEAP group, that are joining us at the International Society for Bipolar Disorders, and they are presenting their experience of being part of the LEAP, as well as the different projects that they've been working on at that conference coming up. And with that, you can really get them involved quite deeply in what you're doing. For example, offering co-authorship on potential publications. They might not be fully into writing it. Some people absolutely are. They would love to be part of that. And again, that's something that you can offer. It's this back and forth of what you can offer them as well as what they're offering you, as well as making sure that they're acknowledged in things like publications as well. So, how to recruit a LEAP. Now, I will say this is an extremely quick whistle-stop tour. There's a whole bunch of other stuff and a whole bunch of nitty-gritty that you would need to get into with a lot of this, and especially of how you're engaging with your LEAP and things like that. But first thing to know is, what are you even asking for? What are you looking for? What kind of experience do you want? And also, what are you willing to give back to your LEAP as well? So before you even get started, a basic question of, well, why are you doing this? Why do you want them involved? Why is it important to what you're doing? What are you going to be asking them for? And what really are the questions you want to know? But what are you willing to exchange back? Again, this is a two-way relationship when you're doing this kind of involvement. But good places to start are just genuine local support groups. Get out into your local community and find out, are there support groups for things like that? A good way of doing that as well is through charities. So a lot of the times for things like Bipolar Scotland, they have set up local support groups through their charity for people that live with bipolar. You can approach them and see if you can get involved. But there are also things like charities like Rethink or Dementia UK that already have pre-set up lived experience advisory panels that you could potentially engage with. You can also go through your institution, because they might already have connections with foundations such as like the McPinn Foundation, which are really involved in making sure people with lived experience have a voice in research. And then there's always the bog standard social media. Get on out there and just ask, is anyone interested in being involved and sharing their expertise? So you can find out more about how some of our LEAP are involved in different projects. So in the Circadian Mental Health Network, Helios BD and Ambient BD, the websites are there if you want to have a look. Gives you a bit more information on what they've been involved in and things like that. And a big thank you to all of the lived experience people that I have had the pleasure of working with and involving in my research. It's definitely made me a better researcher. And a thank you to you too for listening.
That's great. Thanks so much, Amy. As before, if it's all right, we'll go straight on to Raahat and then we can have questions at the end. Thank you.
Definitely, Emily. I'm just going to quickly share my screen. I hope you all are able to see my screen. So good evening, everyone. I'm Raahat, and I work as a postdoctoral research associate at the Division of Psychiatry at the University of Edinburgh. And today's presentation is really, as Amy said, about co-production and the more practical aspects to actually conducting patient-public involvement or engagement within research. So the title of my presentation, as you can see, is called Nothing About Us Without Us. That really sums up what the essence of PPIE or co-production is really all about, where people, especially the general public or the participants or even specific groups that we really intend on working with, have kind of started this movement of saying that whatever findings we generate should not be without the consultation with them. So I'm just going to give a small kind of a practical guide of how to do that, different ways that I have done before, probably giving some examples and some considerations towards the end as well. So just to start with a bit of an introduction, what really is patient or public participation, or like patient-public involvement and engagement? It falls under the broad umbrella of participatory action research, where our participants are active researchers or active consultants in the research process. It helps to include lived experience of people right from the conceptualization of research to the dissemination of it, which can really help to make findings more translatable to policy and practice. And this helps us as researchers to make our findings more generalizable and translatable to the people we really want to be working with. And that's essentially why PPIE or co-production have really gained traction over the last few years. And as Amy briefly mentioned, even funders are asking for it because it makes findings more relatable and overall research more inclusive and diverse within the general population, essentially. So just going to, and I'm not going to go deep into the theory of it, but just getting into small kind of highlights of where these participation models come from and what do they actually mean. A lot of the times we get confused in terms of what is difference between engagement and participation. So over, say, the late 90s and even over the last few years, things have really gained traction in terms of all the models of participation that have come out. And I've really highlighted the top 3 ones that are widely used in research with either vulnerable people or young people as well. So starting off with the Wilcox model, it talks about different levels of participation, right from decision-making, involvement, consultation, co-production, active involvement in research. So it really highlights the difference between, or tries to dismantle the difference between a researcher and a participant in terms of positionality and power, which is one of the most standing features of what conventional research kind of ends up doing, where we end up alienating participants so much by having that hierarchy between us and them. It often creates a sense of alienation and exclusivity, which PPIE or co-production models really try to take away from. Moving on to the Hart ladder of participation model, which is widely used and something that I've previously worked with as well. It really talks about different populations. So say children and young people, their parents or adults who make decisions with them and for them. So it talks about different levels, like on a ladder, on a model of ladder where Level 1 is where we're completely excluding children and young people in making decisions. And level 8 is where we're actually kind of having collaborative consultation and decision-making with vulnerable population and other stakeholders that are involved with them. So Hart really talks about difference between meaningful participation and tokenism, which often lived experience research might end up doing if not done properly. And the most recent one, which has actually been taken from Hart, is the model by Laura Lundy, which talks about the model of participation. And it goes a step beyond to what Hart was saying in terms of meaningful participation in tokenism. It talks about advocacy, rights, voice, generating meaningful impact. Essentially, what is meaningful impact to that specific population that we intend on working with, and how do they make decisions? So these models have really kind of consolidated and solidified evidence in based of using co-production in PPIE methodology in terms of actual and conventional research models. So just to talk about quick methods of what and how to conduct PPIE, Amy gave you a bit of a kind of insight on what we're doing in our research lab regarding knowledge exchange and dissemination. I'm going to talk about how we actually conduct research in terms of using that patient-public knowledge. So we can do it through co-production, where our participants and the members of the general public are active co-producers of research. They might help in co-designing, co-data collection, co-analysis, if they have the kind of expertise to do so. Advisory panels and consultations, which are really the most widely used methods of co-production, where we invite groups with experts by experience to give consultations or give advice on specific aspects of research. It could be recruitment, it could be advisory materials, it could be research questions, it could be some apparatus you might be testing. So it's different elements, and we're just taking the consultation with them. Peer research is something I've previously worked with as well, where we train people or our co-producers essentially to help us conduct or collect data. So we might train them in interview methods, sensitive listening. We might train them in kind of conducting a questionnaire or something of that sort. And that essentially is where that power hierarchy between a researcher and a participant are reduced to the most level, where there are people with similar backgrounds talking to people with similar backgrounds to really eliminate the dissociation between a researcher and a participant. And then is citizen science. And this is quite common these days, especially within health research, where there are a lot of science shops, especially with people who are extremely vulnerable or have very specific experiences, might be marginalized as well, to really train them in understanding scientific methods. And a colleague of mine was actually involved in a citizen science project called the Science Shop here at the Edinburgh University. And they were helping to create like a screening tool for people with respiratory problems. Similar to that is conducting workshops. So it could be workshops for different themes. It could be a form of consultation. It could be like a focus group where you ask different questions and they lead to really helping you map out what your research question, aim, or hypotheses might be. Then is knowledge exchange, which Amy has briefly talked about, wherein we have used our lived experience advisory panel to really help and disseminate the data we have. For ambient, we don't have any data as of now because we've just started as a project. But for Helios, we are really talking about things like seasonality, light perception, use of lithium, and how do people actually feel about their own experience through AI-generated artwork. And that's something Amy has wonderfully explained to you all. And then as collaborations, which are a really important step of PPIE, wherein you either collaborate with the individual or you collaborate with the organization, depending on where you're conducting your research and which aspect of population you're really speaking to. And then is community-based participatory research, where the researchers themselves embed them into the community or come from similar experiences. Say if they are talking about COPD or bipolar disorder or a specific experience of being in social care, the researcher might have a similar experience, and then they are kind of doing a co-production or a PPIE workshop or something of that sort to really understand what do people with that specific lived experience need. So moving on to different stages of PPIE. Now, I've really tried to be systematic with this approach. A lot of the times people ask what really it is and how we can do it, because it is such a broad concept. So we can start out by doing like a mapping or a scoping out. So either you're already working with different organizations and bringing them in, or doing like an in-depth stakeholder analysis, which is really important because often we miss out people who are associated with the people that we're working with. So for example, I'm currently working on a project with bipolar disorder, and often we miss out on experience of people who are suppose caring for them, or people they are caring for. So people in their immediate family, next of kin, children, and they are also the ones who are being directly or indirectly affected by that specific experience. Hence they have a unique perspective of lived experience that researchers might not have. So having that scoping out is really important, working with a third sector organization or a charity, or maybe even the NHS, if that's where your research sits in. Then moving on to the conceptualization. So once you've scoped out specific organizations and have developed like sort of an initial conversation with them, you bring them on to really help you to conceptualize your research. Some people have mentioned in this talk before about including people, say, in a grant writing, in a grant application, or asking them or doing like a priority setting exercise, which they would tell us what is actually needed from us to them, where we can really kind of understand what is their priority, if the research questions we are trying to ask are the ones that they actually need or not. And in most cases, they might agree with us. In most cases, they would just give us unique insights which we might not have thought before, especially in terms of how to engage with people when our active research is rolling out, or once we have also got our ethical approvals, and so on and so forth. Then comes on to the collaboration stage. Now, this is kind of broad, but also one of the most important stages, which takes also most time in the whole process. And this stage is quite relational in its approach, at least in my experience, where we are developing a proper relationship, which is a bit reciprocal as well, where something that our collaborators are getting something out of us kind of including that lived experience in that research brings upon. And that's essentially what collaboration means. It could be, as I said, bringing them onto a grant application, or providing them with knowledge exchange talks, or giving them study updates, even volunteering for them, or just providing them any sort of kind of support that they might need as an organization or as a community to kind of develop or have a better quality of life, so to speak. And they really appreciate this input, no matter how big or small that is, which really helps to solidify the co-production and the collaboration process going forward. And last is the feedback. So once you've done all your PPIE work, maybe have collected data from them, have consulted or got their advice on something, it is really important to feed whatever you're doing back to them. And that's slightly different from sharing of results, because we all publish, go to conferences, try to disseminate as openly as we possibly can. But that's different to what this feedback is. And this feedback loop is kind of like the end loop of PPIE or any sort of like patient or public involvement and engagement, where they really need to know what's going on in terms of study updates. It could be just taking your findings to them and asking them if it actually reflects their experience, maybe even training them and helping you to do analysis, which might be slightly tricky, but some projects have successfully done that. So there's always different ways. And this can be determined in the mapping stage, is what people actually want. What kind of feedback would they appreciate? Would it be a report? Would it be a summary? Would it be a talk by the researcher? Or would it just be like an element of consultation given by people who they consider experts by knowledge to experts by experience? So just giving examples of PPIE. So I was involved in two separate projects. One of them I'm currently working on, and one of them was a part of my PhD. So I'm currently working on advisory consultations, and I'm conducting regular advisory consultations with people with lived experience of bipolar. So we are asking them about what are their, say, clinical outcomes, what are the functional outcomes that they prioritize. We are asking them to comment and feedback on our research design, data collection methodologies, how to engage people in a prospective study, which we will start in early 2025. And that's essentially what we're doing, is testing out each and every aspect of the study, taking them and piloting essentially with our co-producers or people with lived experience. And the second was peer research, where I was a part of, say, an organization that provided support to care experienced young people. I did advisory consultations with them, and then we designed a research project together where they were conducting interviews with their peers and I was just supervising them. So I essentially trained them in kind of skills, transferable skills like interview conduction, sensitive listening, confidentiality, debrief, making sure that they are able to have difficult conversations without being distressed or kind of triggered, because it was sensitive population. We were talking about trauma-focused research. So it was really to eliminate a imbalance between an adult talking to a young person. We kind of came up with an idea of doing a peer research where young people are talking to young people and supervised by adults. So these are some of the examples of PPIE work, or co-production as I would broadly call it, that I have been involved in. Just to quickly end my talk, and I'm also aware of time, there are some considerations that we need to make sure that what we are doing in terms of PPIE are actually correct. Otherwise, if it's done wrong, it could really distress the population and create like a sense of mistrust within the research, from the population to the research community. So it is really important to ensure flexibility, because often we don't know what people might want and we go with a specific idea. So it would be helpful to really be flexible in our approach and have some wiggle room in terms of people might say something we might not have thought of. And that's really important to consider. Second is trusted relationships. As I said, a lot of the collaboration work is really relational in its approach. So we need to make sure we're either talking to people our participants trust, or we are doing it in spaces, physical spaces they trust. Instead of calling them to our units, we go to kind of their physical spaces. It might be a room that they constantly meet for a support group, for example. So having those kind of building up those elements of trust are really important. And as I said, reciprocal efforts. So it could be giving them a consultation or a talk or a seminar in return for their lived experience, which really does not match up to the value of the lived experience, but that's something that they really appreciate. And availability of time and resources. As I said, it requires a lot of flexibility. Therefore, it requires a lot of time and a lot of resources in terms of incentives, could be vouchers, financial compensation, merchandise, something that they really appreciate and have like kind of like a certificate of participation, other than just being a part of the study and saying that they were. So these are the considerations that we need to make in order to make sure that the PPIE work we do is actually meaningful, actually impactful, and does engage with the community well. So that was my small spiel on the importance of PPIE and a practical guide of doing it. You can note my email, and if you have any questions, please feel free to ask them. And I'm happy to answer any questions if there's time left as well. Thank you so much for listening.
Thank you so much, Raahat and Amy. That was such a helpful and insightful kind of practical guide. As you say, it's definitely becoming more and more important. And I think funders are increasingly expecting us to have a clear plan for meaningful public engagement. I guess my question, so I work in kind of basic wet lab science, which is not as immediately translatable to PPIE. And so how would you advise people like me to engage with maybe a more diverse patient group, which is kind of those with respiratory disease that's maybe not so well defined? Sorry, I should say, please do put your questions in the chat if you have any.
Is it okay if I go first, Amy? So I have heard a lot of people, especially in wet labs, say this in terms of how do they engage with people from a diverse group. And I think we always need to remember that if there are participants at whatever end of the other side, that's something that we need to really kind of scope out. And I think that's where the whole mapping exercise is really helpful in terms of who are the stakeholders. It could be, I don't know, ethics committees, they are stakeholders as well. So really kind of knowing from the grassroots to the top, who are the people that they would be involved in? Is it practitioners, policymakers? It doesn't necessarily need to be vulnerable population. In most cases, it might be, but that's not the only people that you would be targeting. It could be other researchers, say with specific lived experience as well. I was previously involved in a small fringe project about researchers of color within academia. And that's all of us researchers with, like, I don't know, postgraduate education within a university setting. So that's a specific lived experience, which not everybody might have. So when we talk about lived experience, the good thing about it, it is so broad that anybody who kind of really falls within that periphery might be, it might be relevant to them as well. And I think that's where the whole mapping is really helpful. And otherwise, as Amy has said, and I have two charities, third sector organizations, they are really helpful in kind of providing you or signposting as well on who might be relevant.
Brilliant, thank you. There's one more question actually from Katie. How do you balance asking patients in public what they would like to be researched versus what's possible, trying to avoid disappointment and not meeting expectations?
So Raahat, if that's okay, I'll jump in on that one. So I have been part of a priority setting partnership, which is basically of asking the public, people with lived experience, even clinicians and anybody that supports people with lived experience of mental health, disrupted circadian rhythms and sleep, about what the research should be looking at, what's important to them. And a lot of the time they have fantastically insightful questions, but that's the problem of how do we not raise the expectations? So it depends on kind of like the scope of who you're asking. Are you asking an entire general public survey and all you can do is just give back the feedback later of been like, here's the questions you asked and here's what we can tell research is important, but it's then up to the researchers to take those and make them, even if it's only bit by bit of those questions, if there's only one aspect of that question they can answer, it's up to them to start on that. So just because we can't answer the questions immediately doesn't mean we can never answer those questions. So it's always trying to make sure that their voices are heard, that we know these are important, and we're going to try and make sure that research going forward takes that into account. But even then, if you're doing it in a more smaller things of like Raahat working with co-production, working with a smaller group of individuals, it's a lot easier to have that open relationship and be extremely honest and transparent of what can be accomplished.
Absolutely. And I think transparency really is the key here, because sometimes we really underestimate how much they appreciate these honest communications. So just as Amy said, in my co-production group, we have been really honest about, or really upfront about the kind of questions we can tackle now versus something we might look at later. And for them, that's really helpful to know, because I think people often get so overwhelmed by these broad questions or like group of researchers just coming up and talking to them. So for them, it's really helpful to have kind of those boundaries, which really then enhance the co-production process, because then you are really sticking to something that's important for the project and for the people. And in that kind of limited space, you then ask them what is important to you and how we can make it happen. And often there are different ways to it, but open communication and transparency is the key, I would say.
Yeah, just to, sorry to kind of jump back, but just to connect it back to what you guys were saying about wet lab stuff, with that open communication and transparency, there's quite a disconnect between things like, even from a researcher standpoint of like wet lab science and then the people that eventually get it translated to at the end. But going and speaking to people with lived experience, they want to know everything and anything they can about their condition most of the time. And if that is just, here's what we're looking at a cellular animal model level to try and understand this one little bit, they want to know. They want to know everything and anything. So don't be discouraged if you think your research is too far down the pipeline. They want to know.
Brilliant. Thank you both so much. Sadly, we'll have to leave it there in the interest of time. So I just would like to thank all of our speakers and panelists again for a really fascinating drive through all the different aspects of kind of strategic research. Finally, if you're interested, we have an increasingly active ECR group within the Association of Physicians, and we'd be delighted if anybody is interested in joining us or learning more about what we do. And I'd also encourage you just to look out for more details about our annual meeting, which is going to be in London in April next year. We always have an ECR session as the first session of the meeting. Last year, Helen and Karen organised an absolutely brilliant session in Newcastle, and I'm sure that this year's will be equally brilliant. It's a great opportunity to meet other ECRs and network, and also to submit an abstract and have a poster or a spoken presentation. So keep an eye on the Association of Physicians website. And with that, I'll say thank you all for attending and to the speakers, and enjoy the rest of your evening. Thank you.